Hi everyone, my name is Aiman Mirza and I'll be presenting my summer research project that was focused on the cultivation and metabolic characterization of CPR bacteria in groundwater. Groundwater exhibits a diverse array of microorganisms that have evolved unique survival mechanisms within energy-limiting conditions. Competition confers a selective advantage to those microbes that can synthesize secondary metabolites, which are specialized biomolecules that mediate their environmental interactions, for example, by functioning as self-defense compounds. This project focused on the bacterial group candidate phyla radiation, or CPR. CPR is an uncultivated group of ultra-small bacterial lineages that has only been defined through the lens of bioinformatics. They present minimal biosynthetic capabilities due to their reduced genomes and are predicted to rely on a symbiotic interaction to meet their metabolic needs. Thus, CPR may harbor an unexplored chemical diversity of bioactive compounds with relevant biotechnological applications. The purpose of this project was to characterize CPR by defining its symbiotic interactions with groundwater microbes and gauge its metabolic versatility by its secondary metabolites using an experimental and computational approach respectively. The experimental approach involved the cultivation of CPR in various growth conditions. A CPR-rich sample was first isolated from groundwater and inoculated in liquid-rich media in efforts to obtain a pure CPR culture representative. Co-culture experiments relied on diverse bait organisms that had been isolated from groundwater. The bait was then subject to various conditions in liquid medias or on plates to selectively engage in a symbiotic interaction with CPR. The centrifuged, lysed, and plate-grown samples were transferred to water media with CPR to see if the absence of growth substrate can elicit a physical or non-physical interaction between the microbes. Visualization. So to determine experimental success, visualization of media cultures was performed using a light microscope to determine the morphological characteristics and activity of both the bait and CPR. Furthermore, samples depicting high densities and the presence of morphologically distinct organisms were sent for sequencing. Of interest was the pure culture of CPR grown only in the presence of growth media. The video here depicts active microorganisms with a circular morphology. In terms of the co-culture experiments, sample microscopic images are provided. The centrifuge samples reflect morphologically distinct microorganisms coexisting without supplied nutrients as shown by the arrows. The lysate sample depicts circular morphologies similar to that observed in the CPR isolate sample. Computational. The computational approach focused on determining the secondary metabolites of CPR, the synthesis of which is controlled by biosynthetic gene clusters, or BGCs. This part focused on analyzing the distribution of BGCs among groundwater CPR genomes recently published in a paper. The first step was to identify the biosynthetic gene clusters by the algorithm anti-smash. The program anti-smash was ran on all genomes and the data was filtered, which revealed BGCs in 40 of the 206 identified CPR genomes, with the highest abundances in the 3 lineages identified here. The next step was the analysis, which involved identifying patterns of BGCs by visualizing its distribution among CPR lineages. This was done by creating a heat map reflecting the BGCs across various CPR classes. I've highlighted 3 classes of interest that are predicted to produce distinct BGCs in high abundance, reflected by the dark purple color. This suggests the diversity of potential secondary metabolites found among CPR that may serve critical for its functioning. The cultivation approaches utilized in this project reveal the presence of active microorganisms either coexisting or found in a pure culture sample. The next step relies on the sequencing data that is yet to be back. The second goal was addressed by determining the BGCs that are present in various CPR phyla, but further analysis is required to confirm the presence and function of secondary metabolites. Thanks for listening.